Now let's see the synthesis of the T3 T4. When we talk about the T3 T4, we all know that the basic constituent of the T3 T4 is the iodine, and T3 T4 they are produced from the cells present in the thyroid known as the follicular cells. When we make a follicular cell like this, okay, so inside the follicular cells of the thyroid there is a substance called as colloid. Okay, this is known as colloid. When we see about what is important about this colloid is that this colloid contain a glycoprotein. This colloid contain a glycoprotein called as thyroglobulin. This thyroglobulin, it is the main constituent. We will see later on how this thyroglobulin is used and how important it is for the synthesis of T3 T4. Okay, so let's see what happens in this follicular cell and what is the role of iodine and how it is trapped and how the T3 T4 is produced. Okay, now when we talk about the thyroid, let me zoom it here. This one thyroid follicle. Let's see, this is a thyroid follicle and this is the blood vessel. You may be familiar with the concept that what is the arteries bringing blood vessel, bringing blood supply to the thyroid. Okay, so it may be, you may be familiar about the anatomy. It is the recurrent laryngeal muscle, recurrent laryngeal artery, laryngeal muscle. Okay, so it is now, we will discuss like it is a blood vessel containing the iodine. So this iodine is not present in the form of I2, that is molecular form. It is present as the ionic form of I negative, and this I negative ion, that is iodine ion, it's the ion negative containing negative charge, is taken into this follicular cell by transporter called as, this transporter is known as NIS, that is sodium iodine symporter. This channel, this sodium iodine symporter will trap the I negative ion into the follicle. Okay, let's see, it is inside the follicle now. Actually this is the membrane of the follicle. Okay, this is the membrane, cell membrane, and this is the wire cytoplasm we are talking about. This is the cytoplasm containing the colloid. This process of the iodine trapping here from the blood vessel into the follicle is called as iodine trapping. I negative trapping. This process is called as I negative trapping. This iodine will go into the follicle by using the another transporter called as CLI antiporter. Antiporter, you may be familiar that symporter is towards the same side and antiporter is the outside. I mean two different sides. Okay, why it is called antiporter? Because iodine will go inside and chloride will go here in opposite side. That is why it is known as CLI, that is chloride iodide antiporter. This is known as chloride iodide antiporter. So what happens here in the colloid, there is one enzyme. Okay, you should remember it, iodine I negative. This is called as TPO enzyme. this TPO enzyme will convert I negative into I2 molecular form. Okay, molecular form, and this I negative I2, that is iodine molecule, will be converted into T3 T4. We will see how it is converted in later part of the discussion. And this T3 T4 will go back into the follicle to the process of endocytosis and will be released into the blood vessel as T3 and T4. Okay, now let's see this step here, how this iodine I2 and how this thyroglobulin, they react with each other and form this T3 T4. Now when we talk about TG, that is thyroglobulin. Okay, thyroglobulin, the thyroglobulin, this reacts with the iodine molecule I2, and this iodine gets attached with this thyroglobulin. Thyroglobulin is a long chain. Okay, it is a long chain containing the amino acids. The main amino acid, which is the tyrosine chain of the triglyceride, is a tyrosine. That is why we say that tyrosine is the main amino acid which is required for the synthesis of the T3 T4. It is the tyrosine, because this tyrosine is a part of this thyroglobulin. Okay, this will get attached, and this reaction is known as organification. This is known as the organification reaction. Okay, now let's say it is the chain. Okay, chain of the thyroglobulin. The structure is not important, but it is important that it contains the tyrosine. Okay, which is important. So maybe it gets maybe like 2 molecules of iodine will get attached, or maybe 3 iodine will get attached. So based on the attachment, the number of attached iodine, it's known as the MIT and DIT. This MIT is called monoiodotyrosine, and diiodotyrosine. Okay, monoiodotyrosine and diiodotyrosine. Now this monoiodotyrosine and diiodotyrosine, they will get reacted with each other. Let's say it is the colloid here. This is the colloid, and there are lots of MIT, DIT are formed. So they will react with each other. Okay, maybe MIT will react with MIT, DIT will react with DIT, or MIT will react with DIT. So based on this, we can get that if MIT, that is monoiodotyrosine, react with the diiodotyrosine in presence of the enzyme known as TPO, it will form the mono plus di will form the 3, which is known as T3, and this DIT plus DIT, which is 2 plus 2 di plus di, will form the in presence of the enzyme called TPO. Okay, thyroperoxidase. Okay, and form T4. Okay, so this is how this T3 and T4 gets formed in the colloid, how we discussed here. Okay, this was the colloid. The iodine was getting converted into T3 T4 in presence of TPO enzyme. So this is how that in presence of the okay TPO enzyme, the T3 and T4 will be formed. This MIT and DIT is formed by the reaction of this thyroglobulin molecule with iodine. This thyroglobulin was present in that colloid here, in the colloid itself, which formed here. This is the colloid, this is the follicle, and this is how this thyroglobulin will react with the iodine molecules form MIT, DIT, and in presence of TPO this will react form T3 and T4. So there is a one clinical importance of this phenomena. This organification reaction, which we discussed here, that is the thyroglobulin and iodine react with each other and form the MIT, that is monoiodotyrosine and diiodotyrosine. This reaction is known as organification reaction, and this reaction here, that is MIT react with DIT, DIT react with DIT, form the different T3 T4s, and this reaction is known as coupling reaction. So here about the clinical importance, we will see that there is a one concept that if we give large doses of iodine, the organification reaction, it will get inhibited. Okay, it gets caught here. This organification reaction, it gets caught, that like it gets inhibited. So this a large amount of iodine is present in, this is known as the Lugol's iodine. Lugol's, L-U-G-O-L-S, Lugol's iodine. So what is the main important about this Lugol's iodine? So before the thyroid surgery, if we are going to do the surgery on the thyroid to decrease the size of the thyroid gland, we give this a large amount of Lugol's iodine before maybe 1 week, 10 days, so that this organification reaction will be inhibited, and this T3 will not react with the iodine, and the automatically obviously the size, the T3 T4 will not be formed, and therefore the size of the thyroid will automatically decrease and shrink, and it will be easy to operate. This is the clinical importance of this Lugol's iodine. This was some clinical relation, and this is all about the formation of the T3 and T4. Thank you.